This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. So we can start the class now, I think. Almost all are there. So we'll start the class. So we are in the first chapter, reproduction in organism. So after the last class, we were discussing about asexual reproduction, right? Asexual reproduction is, it's simple, it's nothing but the reproduction without the involvement of sexual process. There is no sexual process in the asexual reproduction, or there is no gamete formation. Then we discussed about vegetative reproduction. The vegetative reproduction, that is also coming under asexual reproduction. And this vegetative reproduction is of two types. First one is natural vegetative reproduction. That means that is happening naturally. And we have already discussed about this in the previous sessions. And the second one is artificial or horticultural method of vegetative reproduction. In the last class, I just gave an introduction to this. So today we are going to discuss about artificial or horticultural method of vegetative reproduction. So let's move to this topic. Next, we are moving to artificial method of vegetative reproduction. What is vegetative propagation? In the last class, I explained all these things. Vegetative propagation is by using the vegetative parts of plants, we are doing the propagation or the production of next generation. That is called as vegetative propagation. And vegetative reproduction, that is not included, or there is no sexual methods. It is only the asexual process or asexual method. And that is occurring in vegetative parts. Which are the vegetative parts? Root, stem and leaves. And there are so many procedures, fragmentation, then regeneration of different parts, etc. All these are coming under vegetative reproduction. But artificial method, what is that? Artificial method of vegetative reproduction, that is done by human beings, or that is not happening naturally.
Divya,
Yeah,
please share the screen. Yes, share the screen.
Screen is shared, ma'am.
No, only this heading only is showing. Your side only showing. The screen is TeenEinstein only showing.
Yeah, I'm moving to the next part is coming, means I'll show the slide. Just I'm giving the introduction.
Oh, thank you. Sorry, Divya.
Okay. Just I'm revising the last class, whatever we have discussed. That's why I'm not showing the slides. Okay, so now we are moving to the artificial or horticulture method of vegetative reproduction. But in artificial method, what we are doing? They are conducting this kind of experiments or artificial methods in lab or in the field under some particular methods, or they are using some special methods for this. And what is the benefit? What is the benefit of artificial vegetative reproduction? Through this, we can produce variety of plants with different qualities, or we can improve the qualities of plants. We can combine different characters of plants or different features of plants, and we can produce two or more plants. So that is the special or plus point of this artificial or horticulture method. Whichever character we like or whichever character is the desirable character, that we can choose and we can combine those characters from a particular group of parents. See, if we like from parent A, we like the height of that plant, and next one, see, we are producing a new variety of mango. Think one is very good in their contents or the nutrient quality, it is very good, like nutrients. In that character, it is very good, plant A. And plant B, which is very good in the number of fruits. Both characters we can combine in artificial method and we can produce a new variety. That is the speciality of artificial method of vegetative reproduction or horticulture method of vegetative reproduction. We are making use of this in the horticultural methods. So we'll move to artificial horticulture methods. In artificial method of vegetative reproduction, there are 4 types: cutting, layering, grafting and micropropagation. So which are the 4 methods? Cutting, layering, grafting and micropropagation. So we'll move to one by one. First artificial vegetative propagation method is cutting. This usually we are doing this in such plants which can grow from a plant part like leaf, stem and node. So such plant parts we can grow into a separate plant. In those kind of plants we are using this method, cutting. This procedure is simple, very simple procedure. It is having the plant part which is suitable. You can see here in this picture a suitable plant part or the plant part which is suitable for a vegetative propagation, that we can cut and we can place that in a moist soil. See here, the plant part, a mature plant part is cutting from the parent plant and we are placing that in a moist soil. And after placing in moist soil, what will happen? After few days, or we need to water the plants and all those nutrients we need to provide, and after that it will develop roots. Normally that will develop roots and that may grow as a new plant and that will perform the functions of a complete plant. And this is the common method. Examples are rows. Normally in your home also you will do the rows. You will cut the parent plant and you'll grow it as a separate plant. In the case of rose, bougainvillea, they are the main common examples. That's why I'm giving this. It is easy to remember. Bougainvillea and rose. So in both we are doing this method, cutting. So what you are doing? The parent plant which is suitable for vegetative propagation is cut. Then it is placed in a moist soil and you are leaving it for few days and you are providing proper nutrients and water. Then what will happen? After few days it will produce roots. Then you can grow this plant as a separate complete plant. Examples are rose and bougainvillea. Next is, then on the basis of the plant part used for cutting and the position, based on the relative position of the plant or the plant part, the cutting is classified into stem cutting, leaf cutting and root cutting. Which part you are using for planting or to develop the next plant? Based on that and the position of the plant part. Based on that, this method cutting, it is classified into various types. Stem cutting is there, leaf cutting is there and root cutting is there. See, in some we are cutting the stem, in some root. The plant part which is used to develop the next plant is root. In some it is leaf, in some we are using the stem. So first one is stem cutting. In the picture you can see It's a cutting taken from the main shoot or the side stem, the stem which you are using to cut, or from the main or the parent plant you are going to cut in this. From that stem you are cutting and using as a part. You can see in the picture, this is the stem cuttings of jasmine, and shoots which are showing high content of carbohydrate. The shoots which are showing high carbohydrate content, that will form faster. Roots will be formed very faster compared to other parental plants which are having high carbohydrate content, that will form faster. Which part it will form faster? Root will be formed very faster compared to other plants. Carbohydrate content should be high. Those kind of plants or plant parts will form roots faster. And there are different types of this stem cutting. Which are they? Hardwood cutting, softwood cutting, semi-hardwood cutting, and herbaceous cutting. So just remember this: hardwood cutting, softwood cutting, semi-hardwood, and herbaceous cutting. All this just for your neat purpose. There are different types of stem cutting. Just for neat exam, you remember all these. No need to read in detail. Just remember these points. All these 4 types you need to remember. These are the types of stem cutting. They may ask question: hardwood, softwood, semi-hardwood, and herbaceous cutting. These are the methods of stem cutting, different stem cutting. Just for neat purpose, you need to remember this. No need to read in detail. So what we are doing in stem cutting? The cutting taken from the main shoot or the side stem of a plant. That we are using for stem cutting. That we are growing as a separate plant, and the plant part with high carbohydrate content, that will form roots better than other plants. So there are 4 types: hardwood, softwood, semi-hardwood, and herbaceous cutting. 4 types of stem cuttings are there. Just remember that. Then next one is leaf cutting. Leaf cutting, you can see this is nothing but the propagation of leaf bud cutting. This is the propagation of leaf bud cutting. That leaf bud cutting we are making into a big plant or a normal plant. Only that is done in this method. The leaf bud, we are developing these leaf bud as a separate plant. That is done in leaf cutting. So when we are using this method, it's useful mainly when planting material like shoot or some other parts which we are using, that material is less. Only in such cases we are using this leaf cuttings. Where planting material shortage is there, in that case we are using the leaf cutting. There is a shortage of planting material, then we can propagate from leaf cutting, and this is helpful in such conditions. So where there is a shortage of planting material, we can use this leaf cutting method. So in this, each node of a leaf can be used as a cutting. You can see in the picture, each node of a leaf can be used as a cutting. And leaf bud cutting preparation, that should be done during growing season. See, when it is growing, only in that season this leaf bud cutting. For that we need to prepare. Because why we are preparing only in that time? The bud is entering into a dormant stage, means it is very difficult to bring into active stage. If that bud, it is going to a dormant stage, then it is difficult to bring it to the normal active stage. So during the growing season or the growing time only, we need to prepare this leaf bud cutting. So that is the main important thing. Compared to this root cutting and stem cutting, leaf cutting is quite difficult because like the other part, a shoot, that is very easy to develop, and even roots also easily we can develop. But the leaf part, it is difficult to maintain that conditions. So the leaf cutting, that preparation should be done during the growing season. By chance it is entering to the dormant stage, it is very difficult to bring into the active stage, or difficult to bring into the normal active stage. Why? What is the reason? If it is in a dormant stage, rooting won't take place. It is very difficult to get the rooting stage in the leaf cutting. In the leaf cutting, it is very difficult compared to the other method. In leaf cutting, root formation is quite difficult. So if it is in a dormant stage, it is very difficult to form roots or bring back to the normal active stage. It is quite difficult. So root formation will be very difficult in this stage. So we should not do it in the dormant stage. Leaf bud cutting preparation should be done during the growing season only. Examples are cactus and begonia. These are the examples. Then next is root cutting. In root cutting, this is also simpler method, and it is cheaper also. This is a cheaper and simple method of vegetative reproduction. In some plants, you can see in some plants, the other mode of cuttings like the stem and leaf cutting, they are difficult. That means in that kind of cases, we will go for root cutting. So generally, in which kind of plants we are doing? Plants which are producing suckers. Such type of plants, we are choosing this method. So suckers, in such kind of plants, it is easy to propagate by root cuttings. So suckers are the root portions. So in that kind of plants, it is very easy to develop by using root cuttings. And the commercial propagation of blackberry and raspberry. So what we are doing? Propagation by means of root cutting. It is also simple and cheap method of vegetative propagation. This is the simple method and cheap method of vegetative propagation. When the other methods are difficult in some kind of plants, in that kind of plants we are choosing the root cutting method, where shoot and leaf cutting methods are difficult or it's not possible. In such cases we are using the root cutting methods. So generally the plants which produce suckers, in such kind of plants we are using root cutting methods because it is easy to propagate by root cuttings. So in which and all you are using this? Blackberry and raspberry are for commercial purpose, they are using this method. In banana, root cuttings you are using, right? The sucker only you are using. So is it clear? So that's all about the first method, cutting, which is coming under artificial or horticulture method of vegetative reproduction. So is it clear? The cutting method, leaf cutting, stem cutting and root cutting, all these three I have explained. Hope it's clear. Next, we are moving to the second method of artificial vegetative reproduction. The second method is layering. Layering is the second method. So this method, it is similar to the natural asexual reproduction method. Of which plant? Of runner plant. It is similar to the asexual reproduction method of runner plant. You can see in the picture. See, a runner plant will develop in the same way. So this is somewhat similar to the reproduction method of asexual reproduction method of a runner plant. Which one? Layering. See in this picture, from this itself, it's clear how this method is done. So what we are doing in this method is, we are burying a young stem which is attached to the parental plant. You can see a parental plant here, and this is the stem which is attached to the parental plant. We are burying this stem. This is a young stem. That one we are going to bury, or burying of this young stem. So in this picture, you can see a parental plant, right? And there is a young stem also. This is the young stem. And this young stem is produced from the parental plant. So what we are doing in this method, in this, without making any injury, we are bending that young stem. You can see. We are not making any injury. We should not make any injury. We need to just bend the young stem. And this young stem, while selecting the young stem, it should be low growing. From down part, you need to select the young stem, and it should be flexible. It should not be so stiff. So these are the two things you should remember while selecting the young stem from the parental plant for layering. So what we are doing, we are selecting a young stem from the parental plant which is low growing and flexible, and without making any injury, we are bending. We are just bending that young stem which is attached to the parental plant. Now we are not going to separate it from the parental plant. So first we are just bending without separating from the parental plant. We are just bending the young stem to the ground. You can see, just bending to the ground. Then covering the plant part with soil. Next step is, already we have bent this one. Next we are going to cover the plant part with soil. Not completely covering. Instead of that, what we are doing, leaving the remaining 6 to 12 inches above the soil. We need to leave 6 to 12 inches. Then we are just that bent portion we are covering, and we are leaving some part of plant above the soil. Then the tip of that part or the branch, that we are going to bend in a vertical position. So you can see the tip of the plant part or the branch which we have selected, or the stem part which we have selected, that we are going to bend in a vertical position. And we are then what we are doing, and we are starting in a place that we are tying like this. Okay. Then this bending, you can see the bending which is under the soil, it will induce rooting. So what and all we have done? So layering, it is a method similar to natural asexual reproduction seen in runner plants. You can see the same asexual reproduction method in runner plants. They will bend towards the soil and it will produce roots. That is a common method of runner plants, or their asexual reproduction method. So this is somewhat similar. This layering is somewhat similar to that. In this method, young stem which is attached to the parental plant, it is what we are doing, we are burying that part. First we are bending it without making any injuries to that part. We are bending that, then we are covering that bent portion with soil. And while selecting the part, we need to make sure that it is a low growing portion and it is flexible. So such a part we need to select and we need to bend it. Then we need to cover it with soil. Then the tip portion we need to tie. We need to tie with some stick or something just to get a support. Then if you are leaving it for few days, rooting will start. Then this young plant, that we can separate the young plant from the parental plant, and we can grow separately as a new plant. This is mainly done in jasmine and bougainvillea. Examples are jasmine and bougainvillea, then climbing rose. That is also an example. Then different types of layering are there. In layering itself, different types are there. Simple or tongue layering, then compound or serpentine layering, then tip layering, mound layering, and air layering. There are 5 types of layering. In layering itself, different methods are there. Simple or tongue layering, compound or serpentine layering, then tip layering is there, mound layering is there, and air layering is there. Next, we are going to learn each type in detail. So first, we are moving to simple or tongue layering. See, look at this picture. From the picture itself, it's clear. As I explained in the general procedure of layering, in simple or tongue layering also, we can just do the bending or bending a flexible and low growing branch towards the ground. You can see in this. Just bending a flexible and low growing branch towards ground. And next what we are doing, next we are going to cover it with soil. Again, the same procedure, we are going to cover it with soil. So while covering, we need to leave the growing tip of the young stem above the soil. The growing tip of the stem, that we need to leave above the soil, and we should not cover the tip of that stem. Okay, we need to leave the tip of the stem above the soil. Then after few days, in the buried portion of stem, root formation will start. Here you can see the root formation. In that buried portion, root formation will start after few days. And after few weeks, we can check the root formation, whether it is proper or it is sufficient or not. If it is sufficient, what we can do, we can separate that part from the parental plant and we can grow it as a separate plant. So we can do this method in rhododendron, then climbing rose, etc. Example for simple layering is, in which other plants we are doing? Climbing rose and rhododendron. These are the example for simple layering plants. So what we are doing, this is the same as the procedure of general layering. So in this, what we are doing? Just bending the branch, or a flexible and low growing branch towards the ground. And next, we are going to cover it with soil. And while covering, we need to leave the tip portion above the soil. We should not cover the tip portion. And after a few days, the buried portion that will start inducing roots. And we can check after a few weeks, we can check whether the root formation is sufficient or not. And if it is sufficient, we can cut the part or the parental part from the, we can cut the newly formed part from the parental part and we can grow it as a separate plant. So which are the examples? Climbing rose and rhododendron. These are the example for simple layering plants. In these plants, we are using this method, simple layering. Next method is compound layering or serpentine layering. See, there is no need to by heart all these things. Just remember the picture and learn the examples. From the picture itself, you can write. The procedure is similar. There is slight variation in each method. That's all. The next method is compound layering or serpentine layering. And this is also similar to the simple layering. The only difference is that flexible branch which we have selected for layering, that will be, this is the flexible branch which we have selected, right? So that will be covered with the soil in multiple locations. See, the same branch, first we have covered here with soil. Again, we are bending one more time and we are covering with soil. So in multiple locations, we are covering that with soil. And here, after covering one time, one part is exposed to air, or it is above the soil. Again, the same thing, we are bending here and the tip portion is exposed. So the difference from the simple layering is the flexible branch which we have selected, that will be covered with the soil in multiple locations. That's the one and only difference. That means the branch we can bend many times. At each bending, we can cover with soil. So under each covering, rooting will start. See, here also root formation will happen, and the next bend also root formation will happen. So from the same branch, from the single branch, we can make many plantlets. That is the plus point of this. And this method is mainly used for creepers or plants with longer branches. This is used mainly in clematis and grapes, such things or such plants, these are used, these methods are used. That's what I said, just by heard the examples, rest and all. Just learn the concept and write in your own words. It is easy to write. So for board exam only, they'll ask to explain. So it is very easy. Just learn the concept and write it in your own words. See, in compound layering, what they are doing in compound layering or serpentine layering. See, from the title, serpentine layering, just like a serpent, the form, the shape is like a serpent. Or compound layering, multiple times it is layering is done. Just think in that way and write. The branch we can bend many times, and at each bending we are covering that bending with soil. So under each covering, root formation will happen or rooting will start. And from the same branch we can produce multiple plantlets. And we are using this for creepers or plants with longer branches. And the examples are Wisteria and Philodendron. Heartleaf Philodendron, not the normal one, heartleaf Philodendron. Then Wisteria, these are the examples. Hope it's clear. The next method of layering is tip layering. From the title, it's clear that tip portion we are going to layer in this. See the picture first. Here you can see. In this method, first what we'll do, we are going to dig or digging a hole which is having the depth of few inches. In few inches depth, we are going to dig the soil, or we are going to make a hole which is having the depth of few inches. Then from the parental plant, we are going to select a branch. And this branch, we are going to insert the tip of the selected shoot or branch. Where we will insert? In that hole. Then we'll cover it with the soil. So first we are selecting a branch, and here in the soil we are digging and making a hole of, that hole is having few inches depth, and next we are inserting that branch into that hole, then covering it with soil. Then leaving for few days. Then these tips will grow continuously. We have inserted the tip, so that will grow continuously and it will bend upward. This will bend upward. In this direction it will bend, and upward direction it will form. Then at the bend, where it is bent, in that portion root formation will take place. So where root formation will happen? where it is bent, in that place root formation will happen. So we are selecting a branch and we are digging a hole of few inches depth, and then we are inserting the tip portion of the shoot and covering it with soil, and that tip will continue to grow and it will bend upward. This tip, whichever we are inserted here, that tip will grow upward. See, you can see this tip grows upward, and the bent portion, where that bent portion is coming there, because we are burying the tip like this. So the bent portion, their root formation will take place. Then for which kind of plants we are doing this tip layering? Raspberries and trailing blackberries. For such kind of plants, we are selecting this tip layering. The tip part or tip portion is layered. The tip portion, we are placing or inserting the tip in the hole and it is bending upward. Then in that bend, root formation will take place. Clear? That is called as tip layering. Next type is mound layering. This is also another type of layering, and it is also called as stool layering. Stool layering. Note down, it is also called as stool layering. And this is mainly done for plants with heavy stem. And they should have close branch. Excuse me. So they are having heavy stem. For such type of plants, we are using this mound layering. And we can do the same thing for rootstocks of fruit trees. For fruit trees also we are doing, mainly for the rootstock of apple trees and such type of trees, we are using this mound layering. So in this method, in mound layering, what we are doing, in the dormant season or the inactive stage will come for such plants. So in the dormant season, the plant is cut almost 1 inch above the soil. So almost 1 inch above the plant, they'll cut the plant almost 1 inch above the soil, and then covering the base of the new shoots, with soil. In the next growing season, new shoots will appear, isn't it? So first, what they'll do during the dormant season, they'll cut the plant almost from the down portion. They'll cut, just they'll leave 1 or 2 inches above the soil. In the dormant season, they are doing this cutting. So when the next stage or next season arrives, new shoots will form. So just at the base of that, they'll cover with soil. So here you can see new shoots are formed here. This is simple layering. This is mound layering. In the picture, you can see the second one is mound layering. This is just simple layering. In the mound layering, what is there? New shoots. New shoots are formed here. So just below that or up to that portion, by leaving the shoots above, they'll cover with soil. Then enough root growth will happen. If we are covering with soil, root formation will start, and root formation will happen in a sufficient way, or sufficient root formation will happen. And after enough root growth, we can separate each part and we can form multiple plants. So what we are doing in the dormant season, we are cutting the shoot, and after cutting the shoot, we are leaving up to the next season. So when next the good season or normal season comes, it will form the shoots. So leaves and stem parts will develop. So by leaving that portion above, we are covering the down part with soil. So after properly covering with soil, you can see the covering here. Root formation will happen. When the root formation is sufficient, we can check if the root formation is in sufficient form, we can separate this plant part and we can form multiple plants. So the propagation of gooseberries, quinine, etc. All these things can be done by this method. Quinine, gooseberries, then I said apple. For fruit trees like apple, we are using this method. So in mound layering, dormant season, we are cutting the plant at ground level and cover the base of new shoots that appear in the beginning of next growing season. And after there is enough fruit growth, separating the plant into multiple plants. And gooseberries, quinine, sorry, not quinine, quince, different plant. And fruit trees like apple. All these are done by all these are done by mound layering method. Okay, the mound layering is clear. Almost all methods are similar, just few differences are there, just slight differences. Just keep these pictures in mind, then examples also, so it will be very easy to learn. Don't get confused, there is nothing to confuse. Still, just by heart the examples. Then we have done with the mound layering, and next method of layering is air layering. Next is air layering. So in air layering, this is used to propagate large overgrown house plants like rubber plant. Hope you know the rubber plant. So these are used for such plants. And then for woody ornamental plants also we are using this method. Woody ornamental plants that also cultivated by using air layering method. So look at this picture. In the picture you can see in aerial shoot of plants we can do this. See the aerial shoot. Aerial shoot of plants we are doing this. First what we need to do, we need to make a slit on the stem. Below the node we are going to make a slit on the stem. And that we need to do very carefully. And one important thing is there is slight difference in the procedure of monocots and dicots. We are doing this air layering in mainly in the case of higher plants, right? So in such cases, the procedure in monocots and dicots both are having slight difference. Then after making a slit, next we are going to use a toothpick or wooden matchstick. With that, what we are going to do, we are going to open the slit. First, what we are doing, we are making a slit. Then with the help of a toothpick or matchstick, wooden matchstick, we are going to open the slit. Then next step is placing the wet sphagnum moss around the slit. We are going to place the moss, sphagnum moss, which is used in the vegetative propagation and all. When we were learning all these things in the first year chapters, 11th standard chapters. That time I explained. While studying about moss, I explained all these things. So, in air layering, what we are doing? With the help of a toothpick, we are opening that slit. Then, next step is placing the wet sphagnum moss around the slit. That will maintain the wet condition. Then covering it with a plastic wrap. We are going to cover it with the plastic wrap. Then in this slit here we have covered with sphagnum moss and a plastic sheet is there around that. Their roots will or rooting will start in that region. Through sphagnum moss, rooting will start here. In the second picture you can see that. Like this we need to cover with sphagnum moss. We are going to cover that slit. Then we are going to tie a plastic sheet around that. Why? To prevent the drying of that moss. So that wet condition will be maintained inside that plastic sheet. So inside that wet condition, rooting will start. Then what we can do, we can cut this. Here root is there, above shoot is there. So we can cut that plant below this section and we can create a, or we can plant it as a new plant. So in air layering, it is used for plants that form aerial shoots, which are forming aerial shoots. Such kind of plants we can use for air layering. And we can make carefully, first we need to make a slit in the stem below the node. And with the help of a toothpick or wooden matchstick, we need to open the slit. Then next step is, we need to place wet sphagnum moss around the slit and cover it with a plastic wrap. And we need to leave it for a few days. Then after that, roots will start growing through the sphagnum moss. Then we can cut the plant below the rooting section and we can place it as a, or plant it as a new plant. Then examples are camellia, holy mongolia and rhododendron. These are the examples. In such kind of plants we can do this air layering. Other than that, we can do in so many plants. This air layering is a common method which are producing aerial shoots. So hope it's clear. Next we are moving to, so now we have completed two methods of artificial vegetative reproduction. Which are they? Cutting and layering. Next we are moving to the third method of artificial vegetative reproduction. Which is that method? It is grafting. So the third method is grafting. Why we are mainly using this method, or for what purpose we are using this method mainly? This grafting, it is done to produce different or a new variety of plants that have properties of two different plants. So why we are using? We want to produce a new variety of plant which is having the properties of two different plants. So in the starting I said just to produce a different variety of mango or something, we are selecting two plants which are having two good characters, two features. In one plant, plant A, one feature is good, and in plant B, another feature is good. So to combine these both features, we are selecting the method like grafting. Or through this method, we can produce a new variety. That's what I said, we can produce a new variety of plant that having the best features of two different plants. See from the picture, see this picture. In grafting, what we are doing is cutting the parental plant stem in a particular angle. See, this is the parental plant stem. We are selecting two parents, I said. In grafting, we are selecting two parents. The best features we want to combine from two parents. So, in that, we are going to cut the parental plant stem in a particular angle. See, in both cuttings are different. You can see. In this cutting is done in this way, and here the cutting is different. So in both parents, in a particular method, we are going to cut both parental materials. That is the first step. The upper part, you can see the upper part from one parental plant that is placed in close contact with the lower part of the second parental plant. So these are the two parental plants. The upper parental plant, or the upper part which is from the parent A, that part is placed in close contact with the lower part of the second parental plant. One parental plant we are selecting as a lower portion and another parental plant we are selecting as a upper portion. So before doing this, first we need to decide which plant we are going to select for root portion or the lower portion or the down portion, and this down portion, the parental part which we are going to select as the down portion or the root portion, that portion is called as stalk. This portion is called as a stalk. Then the portion which we have selected for the upper portion for the stem side. This is for the root side, right? And this is for the stem purpose. So this portion is called as a scion. This is called as a stalk and this is a scion. So both we are going to keep in a close contact. Then these two parents which are placed in an angle, here you can see in a particular angle. The cutting is in a particular way. Both the stalk and scion both are having different patterns of cutting. Then both we are going to keep in close contact. And then we need to plaster this together. We are going to tie this together. See, you can see here, like this we need to tie or plaster together. Next, what will happen? Next, the vascular system of each plant start to grow and they fuse at a point of attachment over a period. So, after plastering, the vascular system, the plant, we are providing water and nutrients to this plant, and as usual, the plant part will grow. And in both part, vascular system of each plant part will grow and they will fuse at a particular point. In that part, it will get attached. This takes time, but the fusion of vascular system will happen at a particular point. And thus they form a graft. That's how they are forming a graft. And mainly this method is used to develop better variety of fruits like apple, avocado, mango, citrus plants like orange. Then I said rubber plants, rose, etc. In rose and all, you can see many grafts, especially fruits like mango, citrus, all this, like orange and all, you can see this grafting method. So what is happening? The stalk and the scion, in both we are going to make cuttings in a particular way and then we are going to keep it in a close contact. Then covering should be done, and after or over a period the vascular system of both section will grow and it will fuse at a point and thus it will produce a graft. This can grow as a separate plant and that will be having the characters of both plants. Because the upper part is from another plant and the lower part is from another plant. So examples are apple, avocado, mango, then citrus plants like orange, rubber, rose, etc. Hope it's clear. Do you have any doubt in this grafting method? And next, the last method of artificial vegetative reproduction is micropropagation or tissue culture. Tissue culture or micropropagation, that is the last method of artificial vegetative reproduction. And from the title itself, we can make out that this is micro parts or micro portions that we are going to propagate in this. Micro propagation, the micro parts or the small parts which we are going to propagate. Or from tissue culture. From this word itself, it's clear that we are going to culture the tissues. In this method, under laboratory condition, under laboratory methods, from a single parent, you are going to produce plant parts. Means you are going to produce hundreds of new plants or plantlets from a single parent. See, I'll show the picture. This is the tissue culture or micropropagation under laboratory condition. You can't do this in normal condition because you need to make a sterile condition and you need to provide water and nutrients in different proportions. So you can't do this in normal method. This is under laboratory condition. So have you ever seen or ever visited any tissue culture laboratory? You people have any idea about tissue culture laboratory? In tissue culture lab, what they are doing or how they are producing these plants? I'll explain step by step. In this method, In this, we are doing the culturing of plant cells from different parts. See, what is this? Sucker of a banana. This is sucker of banana plant. So from different parts, we are producing many plants. That is what they are doing in the tissue culture or culturing of plant cells from different parts of parental plant. From different parts of parental plant, we are culturing plant cells. So in the first step, what we need to do? We need to sterilize the plant part which is collected from parental plant. So we need to sterilize. For example, we are going to take a sucker of banana plant. In this, the outer part, everything we need to remove, the outer part and the middle portion. Middle portion, see, you can see this part which is collected from the center of this. Here, after cutting and removing the layers, you will get inner portion. That portion that we need to sterilize. By using different type of sterilizers, you need to sterilize. Why we are using this sterilization method? To avoid other microbes. Because we are using this part for tissue culture. So to avoid other infections like other bacteria or some other microbes will be there. To avoid such things, first itself we are doing the sterilization. That is the first step. Cleaning and sterilization of the plant material. Then what we need, we are not going to do with the help of soil or pot or something. Please, students, please wait another 5 minutes. I'll finish up this portion. Then what we are doing? Preparation of a standard cultural media. We are preparing a cultural media, and this media will be containing all nutrients for the growth of a plant tissue. Or step by step, we'll be providing the nutrients. So the tissue will be placed in a specialized medium which is sterilized to form a mass of cells. The media also will be sterilized and the parental plant part also will be sterilized. And this mass of cells which is produced in the sterilized medium, it is called a callus. The mass of cells which is produced inside the sterilized medium, it is called as callus. You can see these are the different stages of tissue culture. And then this callus which is grown in a special medium that need hormones required for their growth. So first we need to add the shooting hormone. First we are adding the shooting hormone. Then see in this tubes, shoots will be produced. And next we are going to add the, after shooting hormone, we are going to add the rooting hormones in the next stage. So here in this picture you can see the roots which are in this media. The root is developed. Here only shoots you can see, there is no roots. But after adding the rooting media, roots will be produced. So once the callus grows into a healthy plant, see here it is a healthy plant now. Root and shoot both are there. So we can move this to, or we can transfer this to a nursery. Or we can transfer this to a favorable condition. Then also we can move this to a field condition, only to a favorable condition where we can provide the healthy situation. And then the plantlets we are allowed to grow in the nursery. Only in nursery condition we can do this. Slowly we are making them to field condition, or we are making them in a hard form to adjust with the field condition. So examples are oil palm, banana, date plant, pineapple, tomato, etc. All this we are culturing through tissue culture method. So from a single plant part you can produce thousands of plants. See, from a banana sucker, if you are placing this in a soil, we will get only one banana plant in the next season. But if you are using tissue culture method, instead of one banana plant, we will get thousands of plants. See, from the single plant part, you will get so many plants. That is the speciality where the stock material or the main plant material is less, or the plant material is not available for such scarcity or such scarce condition. You can use this plant part for tissue culture. You can do this tissue culture, and from that so many plants you can produce. See, in this what we are doing, first we are sterilizing the culture, then multiplication through repeated subculture. We are doing so many subcultures. And in third step, transplanting, root and shoot formation will happen. Rooting hormone and shooting hormone will add, so that root and shoot will develop. Then after forming a complete plant, we are hardening the plants, or that stage is called as hardening stage, and that procedure is called as acclimatization. Acclimatization we are doing, and from this you can select a good plant and keep as a stock plant for next tissue culture work. And after hardening, we are moving this to field condition or greenhouse condition, so that we are getting so many plants. So that's all about asexual reproduction. So hope up to micropropagation or the tissue culture it's clear. Next is sexual reproduction. In the next class we will discuss about this. Do you have any doubts? Any part I need to repeat? Is it clear up to this? Do you have any confusion in tissue culture methods you want me to repeat? Okay, clear. What about other students? Anyone is having any doubt means you can ask. Deepa, Madhumita, Shabana, Vaishnavi. For everyone it's clear. There is no doubt. Okay, just go through this portion. We'll meet in the next class. Hope this is clear. Okay, then we will see you on Monday. We'll discuss about sexual reproduction in the next class. Thank you, students. Thank you so much.